It's my pleasure and honor to introduce Kai Miller, who's our visiting professor. He is now a professor of neurosurgery at Mayo Clinic. I first met Kai in 2003, 2004 as a very enthusiastic MD PhD student. He was going to get 2 PhDs, develop an entirely new field of mapping the brain, and physics of the brain, and great surgeon. And that's exactly what he did. So no further introduction is needed. He's had work that's gone everywhere from the clinical impact of pediatric epilepsy, fundamental physics of brain dynamics. H-index already is older than its age by a little bit. I'll just leave it at that. You're going to hear a lot about how he's used the advances in our technology and in our clinical work to change our thinking, fundamental thinking about the brain. That's all any of us can ask for in our careers. So Kai, also traveling on this vacation, I get to have dinner with them on Wednesday. His wife Dora, who he met during one of his research tours in the Netherlands, very beneficial time. And then his energetic and actually brought his kids back to Auto.
Anything about the kids?
Fantastic. Welcome.
Jeff's their godfather, so. Thanks. Yeah, so Jeff is my son's Otto's godfather, and it's been one of my close friends and mentors for now going on half my life. So a really big honor to be here. I'm sorry that the first speaker we have in this room is me. Woefully inadequate, but you know, I'm very proud to get to come back here. It's really, for me, this is my my home and where I learned to be a scientist and a doctor, and I'm very grateful to you guys for bringing me. I'm Dr. Adam Logan, my 12 and mentorship over the years. What I want to talk to you guys about today, a lot of the beginning of the talk is really stuff that's an extension of what we started when I was Jeff's grad student, and LGMDs and Jeff Rouse graduate student here a long time ago. I don't have any financial disclosures if anyone from the industry wants to be money on it, but no one does. So, yeah, a lot of great friends.
Can you up the voice a little bit?
Yep, I have a lot of great friends here in the department who I get to interact with fairly frequently. Is a close friend that I call often with issues like functional neurosurgery, and also a friend that I spend time with. I thought this photo was pretty funny. I hadn't seen Jeff for a few years, and when he was the head of the ASPN, president of ASPN, we had the meeting, and I ran into him. Same shirt, the same pants, not color coordinated. The apple doesn't fall so far from the tree, I guess. That was that. So I'm going to talk to you a lot today about stereo EEG, and it's important to place science that we do when we're doing science or when we have some technology in the clinical context that it happens in. So when somebody comes to be evaluated for epilepsy, there are 3 main pieces of information that we have. We have the imaging that we can get, so it's early MRI, but also includes PET, and the MEG is sort of a cross between electrophysiology and imaging. Electrophysiology is the second piece that we have of information about the patient. Those are the changes that you see on EEG before going in after seizures. And the third piece of what we have is what you'll hear called semiology. What that means is the symptoms that patients have before, during, and after their seizures. And if these 3 things agree, then that can steer our care right away. So by hemispheric, disorganized structure, but it looks like there's seizures that are propagating from one hemisphere to another, and frequent drop attacks, we'll do a hemispherotomy. Right, we'll do a corpus callosotomy. If they have, they had a stroke around the time they were born, and one half of the brain is essentially dead, we do a hemispherotomy. If they have focal but eloquent seizures, so seizures start, say, in the part of the brain that controls the body, place a stimulator, we may do a laser ablation. But these are in the cases where you would have epilepsy, imaging, electrical signals, symptoms, all align. Well, that would be somebody whose hand shakes during the time of their seizures, dysplasia, the contralateral primary motor cortex, and the EEG over the side of the head. It's where the seizures start from most convincingly. But for some patients, we won't have a dream. Those are the ones where we go to essentially down this whole other pathway. I'm not expecting you guys to read all of this, but it's just at the point that we go from this idea that we don't have agreement or concordance between the imaging, the symptoms, electrical changes. So we do implanted recordings with electrophotography or stereo EEG. And I'm going to show you some pictures of what that is. So this is stereo EEG. These are electrodes that are placed with about 2 millimeters of precision using a variety of potential guidance techniques. We use a robot. I think robot is used here at University of Washington as well. And this is what they look like as they go in for the scientists in the audience. There's a bolt that's aligned into the skull, and then wires pass very tactically through those bolts. And you can see that we place them all throughout the brain, including the thalamus now. And I'm going to show you images throughout this talk where we see a brain and then we see activity on axiocoronal and sagittal slices of the brain. What that is we take the MRI pre-implantation, the CT from post-implantation, merge them together, and then from that, slice the MRI and plot brain activity. Anyone who has any interest in papers, what you see in this talk, some of them are preprints, some of them are published. There are QR codes, either to get the papers when they're actually published or to get the preprints on bioarchive server. So please feel free to snapshot those with your phone. There's an image up in the back of the room as well, if that's closer to you. Also, just email me. So when we think about this transition from electrophotography, which was the measurement of electrical changes in the brain from the brain's convexity or surface of the brain, moving from that to stereo EEG, we're now going from sort of a regular covering of the convexity of the brain or the outer surface of the brain to a regular and sparse measurement of the volume of the brain. This allows us to sample deep structures. So this is particularly important for seizures starting, for example, in the insula. But it does mean that every time we place one of the electrode leads, we have to have a specific hypothesis about where the seizures may be starting from or spreading to. Placement of that lead with high accuracy reflects our hypothesis based about where the seizures. We also do a variety of new interventions through this. So a lot of times we do test stimulation prior to putting in a permanent stimulator with stereo EEG, and I'll show you some examples of treating things other than epilepsy with that approach. We can also do things like radiofrequency ablation, so we can actually burn brain through the stereo EEG by passing high intensity electrical current through them. When we think about the implications here for research with stereo EEG, now looking at, and I'll show you briefly what this plot here is called the power spectrum. It basically tells you power as a function. If you look to the left of that, though, you can see that the raw signals, even by eye, are very different than the electrical signals that we measure from the cortex to, for example, the electrical signals that we measure from the thalamus. So we have to understand that there's a difference. Signals and our math that we use has to accommodate that. One thing that is very different is the level of detail that we can get within functional domains in the brain. So a lot of the work as part of Jeff's team, looking at the surface of the brain, the representation of visual areas, and really picking apart the way that outer world is represented in detail. Can't really do that with stereo EEG because we're measuring so much. Again, we can sample deep structures. We can study circuitry much better than we used to be able to with electrophotography because now we're measuring on multiple levels in a hierarchy of information. And then movement area, the area that sends signals out to the body, we're also measuring planning areas and the supplemental water areas, We have to remember, like I told you at the beginning, that heterogeneity in the tissue that we're measuring. There are signals of it. So there's only really going to be one thing I'm going to talk about that has, and that is, like I said, for this Fourier transform. And what that means is that if we have some signal where we're measuring voltage here, down as a function of time, going to the right, we break that signal up by talking about underlying waves of activity, rhythms or oscillations. You hear people use those three words. Loosely speaking, that will mean the same thing. Talk about power at a particular frequency. And when we talk about power at different frequencies, what we mean is that in our raw signal, piece apart higher and lower frequency, and then we can plot those together, bumps at different frequency ranges, so that when we superimpose them, we can put the original signal if we want, but we can also go from saying, well, this is one characteristic power spectrum there, and then when I engage in some activity, in this case it might be moving the hand, you see particular frequency range that kicks in, and that's here in pink. And when we stop moving the hand, it goes away. Okay. And so if we do the most basic experiment that we can do, where we have patients perform movements, we have them stare at a blank screen, start out, measuring activity from stereo EEG electrode that passes through the primary motor cortex. We can measure one characteristic power spectrum, like you see here. So I showed you something before that has peaks at all the frequencies. What you actually see is that there are specific spots where you see peaks, but you also have this general fall off shape where it's like it's sloping down from the left to the right. And that fall off, that we spent a lot of time on here at University of Washington, it's called a power law in the spectrum. But the important thing about it is that if you engage that area by giving a patient, for example, a cue to move their hand, measure, they perform in response using EMG. And then we can look at the electrical changes in their brain, and you see that there are changes across a wide range. That change across a wide range of frequencies, what it means mathematically, model the signals that you measure, and also if you measure from single neurons, is that sort of the average action potential rate of the population of neurons that you're measuring from is reflected by increases and decreases that happen across a wide frequency range. And we'll typically capture those for purposes of this talk between 65 and 115 hertz. But really what that means is if you hear people talk about high frequency or high gamma, capture those changes by just looking at ranges that are between frequency ranges where you have artifact from the lights below. So 60 hertz, 120 hertz, you have noise from the ambient electrical signals that are produced by the power. And just to review that briefly, so you have a bunch of neurons here. This is showing a picture of an electrophotography electrode, but the principle turns out to be the same with stereo EEG. All the neurons that you're measuring from are sort of aligned in their timing and firing. Something that looks like oscillation, peaks going up and down in a particular frequency range, and either be an oscillation, or you may get things that are feed-forward input. So for those of you that are familiar with chemicals and science, probably about later, but clinical world, we have bodies and CCPs. What we measure that's most specific for local function are changes that are across a wide range of frequencies, like you see here. And when you look at the raw traces, those are a little bit harder to pick up by eye because it really is sort of a jitter in the time space. Data and the voltage is measuring, you see sort of really the only time you can see a difference is if it just looks like there's more jitter happening. Not necessarily clear to see it as these other changes that we're just going to skip past this slide briefly, but basically this is just showing that if we perform simulations of what might be producing the potential measurements that we measure with our implanted electrodes, that if we look at correlates of firing rate, like you see here, correlates of action potential rate, they're reflected by across a broad range. And going back to this is just showing that we capture them in high frequency range. They give me a warning. I'm starting to get it.
We got a fan here, so it's really.
I said, I was asking Dr. Coe to give me a warning if I'm running out.
Yeah, I'll.
So I'll raise my voice. So there are a few primary things I want to talk to you about today. If the volume gets down, Jeff, could you just yell at me? Sorry, Dr. Ojman, could you yell at me? First thing we're going to talk about is using functional changes in stereo EEG, much like what I just showed you. And for movements of the hands, the types of changes that you see, but you can also basic functional speech mapping. So I'm primarily a pediatric neurosurgeon, and many of these patients can't participate in stimulation mapping the same way as adults can, which means that you stimulate through the electrodes and you shut down function. See a corresponding change in behavior. So what we do if we can't do this stimulation mapping to shut things down, in fact, we do this in our patients anyway, perform basic tasks, and we look at those changes in the electrical signals in response to those tasks as a clinical tool. I might give a patient a cue here on the screen. That's a picture of an apple. And their job for speech is they say the word apple. And then we can measure changes in the audio as you see there, and we can also measure changes or the signals from the stereo EEG, broadband. All that broadband means is that same high frequency activity that we measure. We can also have them do tasks that are supposed to engage sort of speech association areas. So in this case, we'll do the same thing, but we show them a picture, and a picture maybe of something they eat, and instead of saying what the name of it is, so instead of saying pear, they then say eat in this case, and that verb association tends to recruit the association areas we have when we speech. And plot these out on the surface of the brain in a similar way to what I just showed you. For interesting cases like these, where it's any of the residents want to say something that's abnormal, this is great. This is a radiographic image you see here on the MRI. Anything odd about this patient? The interns, medical students. What's that?
It's on the right side.
Yeah, it's on the right side. So this patient happened to be right dominant. Here. Okay. And and so we have actually these types of changes that we see also correlate with fMRI. Okay, so we do with task-based mapping, and we can use this in cases where you're able to do fMRI, which we can't always do with children. See that the electrophysiology that we measure with stereo EEG correlates with the fMRI. Because we're doing things like radio frequency ablation, where we're permanently destroying tissue where these stereo EEG electrodes are, this can be very important for decision-making when I have my stereo EEG patients implanted. So we do this with each of our patients. It's based on just these simple protocols where we show pictures. They came out of work that Dr. Ojman did in Harborview and at Children's Hospital. And this is just showing that in addition to these classical convexity areas we're used to seeing on the surface of the brain, we can also get speech areas during both noun production as well as verb generation in the motor insula and the classic receptive speech areas like Wernicke's area, areas like the cingulate that may be involved with speech. We can do this in areas that are seizure onset zones. Okay. Even though it's where the seizures are starting from, try to map there with this technique. If you try to perform electrical stimulation mapping, it kicks off the seizure with these patients, so you really can't perform. Task-based mapping works really well for that. Whether it's hand or foot movement, speech production, or any associated task you want. So if you're really interested, let's say in memory, or patients will have on things like ventral rotation and things like that. We've had computer programmers. We just design customized tasks from this basic behavioral tasks with images that relate to that, and we're able to elicit activities areas, even when they're in the seizure onsets. In the injured brain, it also works as long as that area is still computing. So this is a patient who had seizures following an MCA stroke. Number of the stroke for function with this task-based mapping. So now we're going to get to some of the science that we have. So when we started doing this a couple of years ago, my first graduate student, Michael Jensen, he'll be not the Michael Jensen you guys have coming as a spine fellow. He was my co-resident at Stanford, but it's Michael Jensen from Mayo. And he'll be applying as an MD, as a neurosurgery resident. He's my MD PhD student. He's really good, but he started, we're measuring from the volume of the brain, and I wanted to give him the most basic task I could. So I said, well, we've known for a very long time that we have this representation of the homunculus where you have the face laterally, the hand and arm dorsolaterally, and then the leg dorsomedially. We've known this organization since the time of Penfield, 90 years ago. And our measurements of this area have always been the convexity of the brain. Why don't you just do a simple measurement and show that it extends down into the central sulcus and extends down into the depth. And so he went off and he did that. And I just measured EMG from the face, from the arm, and from the leg, while he had the patients perform these basic movement tasks. And if you did see, as you see here with the white arrows, I'll talk about the white arrows in a second, representation of the foot medially and laterally, and the tongue sort of more inferolaterally, as we might expect. But he also came to me and he said, well, there's a problem because I'm also getting these other areas that are active no matter what the patient does. So whether moving the right or left side of the body, moving hand, foot, or tongue, these areas are always active. The only problem is they're sitting down in the central sulcus, but we don't expect this. So I said, well, you probably did do something wrong, but let's hammer down on it. So we did. I sat with him and measured this again with 13 subsequent patients. We started doing the analysis. We had all 5 patients. So another, let's say 8 to 10 patients that he measured with, and we always would see these areas that were active no matter what part of the body we were moving, the patient was moving. And so we're able to generate somatotopic maps because there are somatotopic regions just as we would expect, and these are more strongly represented in the convexity of the brain. Classic Penfield somatotopy can reproduce that with stereoEEG. Here you see sort of color tuning for the hand, or the face, the hand, and the foot. Using this sort of this tuning picture, this is showing you how the different regions of the central gyrus are tuned for different types of movements. I can't tell. From here, I get a lot of glare, but I don't know if you guys can see that. In addition to that, there are also these areas that are active no matter what part of the body is being moved. So we call these areas of shared representation the Rolandic motor association area, for the central fissure used to be called the Rolandic fissure. So in order to sort of preserve that name, an area that seems to be an association area sitting where we never expected to find it, so we call it the Rolandic motor association area. And if we cluster across different patients that we have, I'm not sure how well this video is going to work. Yeah, it's working okay. You can see basically that you do have these somatotopically specific areas for the hand, the foot, and the tongue. And in addition to that, you have this Rolandic motor association area here, red, that sits down deep in the central sulcus. And when we map it, function of depth from the cortical convexity, you can see that the Rolandic motor association area, the RMA area, is deeper than the somatotopic areas are. The somatotopic areas are in black, and the RMA area getting deeper in the brain is we want to know, well, what's this area doing relative to movement? look at the time course of broadband activity. So here you have one of these horizontal bars that you see in the background. It's the time of movement of the hand, foot of the tongue, with the foot in purple, the tongue in green, and the hand in orange. You can see that, and these are defined by the EMG, which you see along the bottom. See that there are areas that are somatotopically specific for the foot or the hand. The timing of activity from those very tightly coupled to the time that they move, which is good and tells us that per area. In addition, you have this RMA site, this association site. It's deep in the central sulcus and it's active no matter what part of the body you're moving. In order to calculate relative timing of anatopically specific areas in the RMA area compared with the EMG, you can just take the two signals and slide them compared to one another and calculate latency. Without going too much into the details of the figure, what it shows you is that both movement of the both activity in the somatotopically specific areas and the motor association area precede movement. Okay, if we try to understand by doing the same overlap, where we have these two traces and we're overlapping them with one another, whether the RMA area precedes or follows somatotopically specific areas, we find that one doesn't really lead the other. So they seem to be acting in tandem. To summarize, we can use stereo EEG for volumetric recordings in the brain. Motor homunculus is preserved into the depths of the central sulcus. You have classic somatotopy, but it's interrupted by this dissociation area, the RMA area. And also we can use stereo EEG as a tool for functional mapping in the clinic before we do things like laser ablation, radio frequency ablation, surgical resection. We also do some of the same kinds of things that you've probably seen here at UW, which are feedback experiments with stereo EEG. So based on a paradigm that we first used at Harborview, my lab performs imagery-based feedback. So that's where you have patients imagine moving their hand, record from a primary motor stereo EEG electrode, and then you capture that same high frequency change. Couple the high frequency power to a simple movement of a cursor on the screen, as you see here, so that patients are trying to move to hit a target that's either on the top or bottom of the screen or the left or right of the screen. And the reason why I think it's important to do basic brain-computer interfacing like this is that when you look in the news now, right, you see all this stuff about Neuralink, you see all this stuff about paradromics. There are all these companies coming out of the woodwork that talk about high dimensional control, where they're doing large craniotomies and sticking electrodes in the brain. And everybody's basically bragging about the fact that we're the first to do control this particular type of device, use this kind of recording algorithm, or we're just a little bit better than someone else, or we're Elon Musk. Basically the selling point of Neuralink, because they're just doing a lot of the same timing stuff has been before. But what I think is really important is not to say, well, can we be the best in terms of getting high dimensional control of devices? So, we have 32 degrees of freedom we extract from some penetrating electrode array. But instead, are there general principles that I can do by having simple feedback experiments that might help guide the placement of these high density arrays in the future, or might help guide distributed brain-computer interfaces where you have large electrodes that are more like PBS electrodes that we implant in the future. And so we really just do basic brain-computer interfacing, where I've shown you here, it's 1 degree of freedom driven off of an area close to primary motor cortex. This is what it looks like for patients. You can see this patient is consented to have his picture shown, but you can see him imagining moving his hand to control a computer cursor to go up and down. So that's on the left of the screen where the computer cursor is. He's just performing hand imagery. You can see on trials where it's difficult, you may see him cheat a little bit by moving his right hand. We do have patients do this both moving and then imagining moving. Reason being that somebody you're trying to restore function to shouldn't have residual function there already. So they're actually trying to move their hand. They're not just imagining it when you have somebody who's paralyzed. Okay, so we have them perform these tasks. We look at learning. So if we compare the power in that electrode that we're measuring from during up and down targets. So up and down is red and blue, lower target is blue, and correct trials are circles, incorrect trials are asterisks. See that over increasing number of trials, patients learn to control the cursor extremely well, and they get very high performance. This guy gets one wrong, but it just means that it takes trial is about seconds, so it can take 10 to 15 minutes to get really robust control for some of these patients. If we have any electrode, regardless of where it is, that's tuned when we do a movement task, have patients get nearly 100% accuracy in this task when we measure from these electrodes in periatlantic areas or pre and post central gyral areas. And when we have them do imagery, what we see is 50% here is chance. We have a fairly high number of patients that have great control, as you see, where it's about 100%, but we also have a fairly high number where they get very poor control. So if we look at control over time, what we find is that if they have, for many of the patients, they're not going to get better with time. Okay, so they tend to do really well if you have electrodes that they're doing an imagery associated, so they imagine moving their hand, they get a cursor to move. Some of them do really well from the start, right? So they get almost perfect control in just a minute or two. But then there are patients where you can go, very far out and they're performing essentially at chance, and that's where they tend to. We're getting feedbacks based on electrodes that probably weren't really in primary motor cortex at all. So that tells us that as people are putting in brain-computer interface arrays, they will put them in areas like the parietal reach region, dorsal premotor areas, ventral premotor areas. The control there is probably never going to be as good as if you put electrodes in primary motor. That's a sort of a basic principle that we get from doing a stupid one-dimensional brain-computer interface task. Because we get so many patients that we can work with, right? We have patients here, with the arrays that you see that are in the news and so forth. That's one or two patients ever in the study. So they never really can say, hey, if we just focus on primary motor cortex, that would be better than these implants that are in other brain areas. And the other thing that we're looking at, since we're interested in these types of principles, is as you get feedback on primary motor cortex, as you form imagery, what becomes active. And so this is a set of axial images showing you activity associated with overt movement of the hand, imagery of the hand, and you can see here with imagery, you start to see sort of dorsal premotor areas get engaged. But then when you get feedback, you can have areas down here, for example, that are active in parietal reach region that are not very strongly engaged during overt movement or imagery, but once you give feedback, they are more strongly engaged. Now, that doesn't mean that that's a good place to feedback on, as I showed you before, but it does mean that as you engage with control of a virtual image or virtual object, in this case, movement of the cursor, start to engage some of the more higher order brain areas associated with movement, and understanding the differences in brain circuits during feedback will be important for understanding principles that you need when you are putting these, 1,000 channel arrays like people are talking about now, where you put them and where you focus them on, since it's always just a small patch of cortex. Right, so this would tell us, well, maybe that if you want to control a device, you put that high density array in primary motor cortex, and if you want to have some kind of control of a device or more nuanced control of the device that involves interplay between brain regions, this is how you put in large electrodes. You identify the areas that interact with primary motor cortex selectively during feedback, and those are the ones you include. Anyone have any questions about that? So I'm going to move forward to other stuff, but I know there's some brain interface people. Right? Yeah. Hey.
Can you start? Is there plasticity that after a while it becomes reconfigured and they can actually control it?
Well, I mean, we know that's true actually from F. Betts, who's here
Yeah.
in the building. He showed in like 1969 you could operate. Seem to see things like that with some more fun. My patients that we're working with, it's an hour to control that. So we don't really know, but we know that at least for single neurons can do that. We know from people like Dan Moran at WashU that identify a particular frequency range that's important in a particular brain area and give a monkey several months, they can learn to control that. But what we do want is we want an intuitive mapping, so it's not, it doesn't require here's some feedback to get control. We want something that works pretty well. And the reason for that being, it's just, you know, at some point you abstract away from hierarchy that you have when you're learning something to low level representation. I'm not sure if the sound is going to work on this. This is just showing somebody playing Space Invaders with this. I apologize. We can get the video to.
It's not as impressive.
Anyway, that would have been fun. Those of you that are interested in this kind of stuff, people here, the Motor Association here, and please do look for Michael. He's stellar, and he'll be an applicant. He'll be an applicant in 2 years to start. So, how are we doing on time? Yeah, you got like. Okay. So I'm gonna just show you something briefly about probing brain networks using electrical impulses. So one thing we can do, and this is a sort of a change of pace, but we can try to understand how different regions of the brain interact with one another, or what the effect of stimulating the brain is. Stimulate one brain area, what's the effect on another brain area, by using something called single pulse electrical stimulation. If you want to map connectivity in the brain between arrays of electrodes you implant, what's a way that you would do that? The way that we do it often is something that people call cortical evoked potentials, or single pulse electrical stimulation. And that's where you give electrical stimulus between two adjacent pairs of electrodes. Then you wait several seconds and then give another impulse of a similar. The reason why that's important to wait several seconds is that you're going to induce a voltage change that you can see in the raw potential. The raw potential, like there's an example of this, SCP is an example of this. But that can tell you whether or not this area where you stimulated and where the area where you measured a change are connected with them. And this has been an area of interest for many in the field. The classic model that folks have had for this is that there should be some shape like this. That's the cortical evoked potential that has some 1 and 2, P1, P2. So this is where the state of the field had been for a very long time in terms of how we interpret these signals. That all of your responses should look like this when you stimulate in the brain, and then people would measure the relative latency of these and the magnitude of these. Say that that should tell us about how areas are. So the issue that we run into is that if we do these evoked potentials in stereo EEG, stimulate, let's say here, pulses that are separated between 7 seconds. We jitter the time so that don't have effects of anticipation in the electrophysiology. And then we measure these responses in different brain areas. And if you give about 10 to 15 impulses, here you can measure the average response to that. And that'll have some particular shape. Here people use the word cortical evoked potential, or CCEP. That's what that shape means. That's what that word means. Problem is if we look, and this is just stimulating at 5 different sites in 2 patients, you get a whole variety of evoked potential shapes. So this variety of shapes here is in no way adequate to. You can't describe this whole variety of shapes by this standard shape that people have said that the response should exist in. So a lot of the work in my lab is to develop algorithms to understand these types of brain data. So if there's no standard shape to think about, how do we? So to do that, we have a set of paradigms that we use, and I'm going to go fairly quickly through this, trying to get to an illustration. But basically, there are a few ways you can simplify this process, because we usually will have something like 1 to 200 electrodes stimulating, and most of them measuring from all of them. So if you have 100 different electrodes and you're stimulating all of them, measuring from all of them, you're then trying to characterize 10,000 interactions. So mathematically, that can be very difficult. So we break down the problem mentally into a set of paradigms. The most simple of these is what we call the hypothesis pre-selected. That's where we pick two brain areas that we think ahead of time because of something we already know about anatomy or fMRI or something like that. And we say, well, we're just going to look at the stimulation and response in this interaction we already know about. An example of this would be, we look at limbic circuitry, we put electrodes into nucleus of the thalamus here, and we can measure responses in the posterior cingulate. We also measure responses that we get from stimulation. Oh, sorry, I put the ANT in campus. This was stimulating the hippocampus, stimulating the ANT. We can say, well, these are the only sites that we care about. We're just going to look at a couple of interactions, and we can do things like quantify the delay of these. So that would be a case of a hypothesis pre-selected. We know limbic circuitry is connected, the latency, and we don't really have to worry about some complex formulation because we know what's going on at a time, or at least we think we do. And we live in this world where our hypothesis is pre-selected. We try to do this thing, which is what has been done in a lot of the field, where you have basically this 10,000 comparisons. It's really hard to make any general statement. So I really just. We also have something we call the divergent paradigm. And that's where you say, well, if I just stimulate one brain area and I measure from a bunch of other brain areas, what do those set of responses look like? So say we stimulate here at the site where you see a little symbol, and we get one typical response. And if we look at the response that we get at a different site, another response, it can be a little bit difficult to understand this, because what happens is that basically the same output type here, because it's going to different brain areas that have different laminar organization, that have different microcircuitry, you can get very different shaped responses, even though it's projecting, let's say, through a long white matter track without a thalamic relay or anything else to a superficial area, superficial layer. You get responses that look very different in shape to one another. So trying to interpret that can be difficult. What can be easier to interpret is what we call the convergent paradigm. That's where you focus in on one site in particular. You say, what's the effect of stimulating other places on this place where I'm measuring from? Here, because you're always measuring from the same patch of brain in the same way, different shapes of the signal in time tell you that there is fundamentally different type of interaction that's happening between that site that you measure from and the place that you stimulate. So in order to address everything except the convergent paradigm, we developed this tool called canonical response. I'm going to go through this relatively quickly, but I just want to show you what it is and why it's useful. So for the scientists in the audience, it allows you to take any evoked response that you have, whether it's an electrical stimulation or anything else, and parameterize it without having to know ahead of time what that response shape should look like. So here, let's say we stimulate at one brain area, measure at another, and you get this set of 15 responses here. We can take the first response, we do something called unit normalizing it, or we normalize that, and then we look at its overlap with all of the other responses except itself. We do that using something called the dot. That gives us a cloud of data, and if this cloud of data is significant to 0, we know we have a reproducible shape there. Rather than normalizing just the first trial, we can actually normalize all of the individual trials, get a cloud of data like we see here, and we collapse all of them. We don't order projections that we've done. Cloud of data here, and if this data is significantly different from 0, we know we have a significant crash. So it's just a very simple tool that identifies when we have a significant response. Problem we can run into that a lot of time we get responses where we may send a second of data in, but really our response lasts just a very brief amount of time, right? So if you look at this here, well, this would be the cloud of data that we got, like I showed you in the previous slide, from projecting trials into one another. And although it's significantly different from zero, we're including a bunch of jump data into our measurement there. Okay, so we can actually use the trick that we came up with. And what the trick is, we shrink the amount of time that we're looking at here. Right, so I can take less and less time, and the average of these clouds you can see actually gets more further and further away from zero the shorter amount of time we look at until we get essentially past where that structure is there. Okay, and rather than doing it at, let's say, full time, half the time, and so forth, we just do it at every sample point or at every 5 milliseconds or so, and then we get a profile. This black line here is the average of those projections as a function of how much data that we sent in here. Then what's cool about that is that this was our average response that we had here. Profile tells us that really the data are only significant to about 80 milliseconds here. So it gives us a generic tool to identify how long a response lasts. And this seems to me like the type of thing that we should have done, 30 years ago when we first had the computing power to do it, but no one really did that. You'll notice that this is very different from the response or the amount of time per response that we would have had. We applied it to that example that I showed you in the previous week. We can quantify difference time for how long these are significant for these two very different shapes from one another. Then we can actually do something called parameterize the shape a bit. And what that means is that we know that some response is significant only over that interval there. A single trial here, we can say, well, the voltage that we measure here in black, what it is it's this voltage is a shape that we get from stimulating. We call it canonical shape here, and that's in red. We can scale it up and down because it may change a little bit from trial to trial. Plus, some residual, think of this residual as noise, or we can think of it as brain activity that's not induced sort of as a normal deflection from our stimulation, so that we can talk about the voltage here as being a characteristic shape that we get from stimulation. That's red here, which is what we call C, scaled, plus some other stuff, and that's what this group letter epsilon. Do that on single trials, not just on individual ones. And the reason why this is, not just on average. The reason why this is important, if you want to say whether or not this is significant, we now have a set of parameters here that quantify things like the signal to noise ratio of a single response, explain variance of a single response, and the magnitude of the voltage just over the significant interval. That's the scaling from this Greek letter alpha. This is why it's important. Okay, so we could have the shapes that look very different from one another like this, and our algorithms immediately tell us that in some of these cases, when I've stimulated and measured from somewhere else, nothing important happened. But in these other cases, we had something important happen. It just looks very, very different from one another. But because we've done this mathematical formulation, we can remove the shape out, even though the shapes are very different. We can do things like compare the explained variance of electrical stimulation in this response versus this response. So now we have a technique where our algorithms can parse very, very large sets of interactions so that you can do that 100 stimulation sites and 100 measurement sites and look at 10,000 interactions with algorithms, get statistics associated with those, rather than having to look at it by eye, say what we think is important. Almost done. I'll show you guys one cool example of this. I'm going to skip. There's a clustering technique for those that are interested. I will talk to you about it after. I can send you papers, and I'm around actually until the rest of the week. Of course, but I'm going to skip past the clustering technique. There's a lot of fun math in that. I know every surgery resident loves. But these characteristic shapes that we get, there are a couple of things that are important. So this is the work of Harvey Wong, who's just like we have pairs or SSEPs, there are characteristic shapes that we get. Sorry, my computer is crashing. There are characteristic shapes that we get when we do things like stimulate in particular region of the brain, like the lateral sulcus, and measure elsewhere. But even more importantly, use this clustering technique and measure from the collateral sulcus. When you see that across people, stimulate in the hippocampus and measure from the collateral sulcus, there's one specific shape for that interaction. Also measure from the collateral sulcus, but stimulate instead insula, get a different characteristic shape. I do not know. Right here. I get the spinning ball on my computer. So that's all this is that if you measure at one site, you get different shapes depending upon what you stimulate. These hold true across patients. That's kind of neat. It says we can start to build up a library of interactions for different areas of the brain. So if you have a device and you want to know are the dynamics that we have, we measure our electrical signals in the collateral sulcus on, let's say, seizure start there, stimulate an insula. Are those dynamics changing as a function of the drugs that we've given or whether the patient's awake or asleep? This gives us a toolkit to do that in. The last thing I want to do, we have time, is to show you how we can use all of these tools for a brand new patient. Dr. Koh is saying we're out of time.
Yeah, I'm going to have to leave. You can keep talking.
3 minutes, okay. So this is a patient who had something called Lance Adams syndrome. Okay, so basically, he tries to do things like engage in movement, starts to get this myoclonic locking up. And we went through a full ethical process with our IRB, with an individual ethics panel, and with collaborators around the country to talk about the ethics of it before we implanted him a year later with stereoelectrodes. We then wanted to identify candidate places to stimulate. So rather than just guess what the right area is and put a permanent UBS system in, we wanted to do what we do for epilepsy and put a stereo array in. So we did that, putting electrodes into the deep brain areas we normally think about, like lobus pallidus, subthalamic nucleus, and the motor input regions, thalamus. And we included centromedian nucleus because he also had seizures. And what we found when we looked at the effect of those on primary motor cortex, these are stereo EEG electrodes, was that basically you could do stimulation for 5 to 6 hours at a target site and then perform the movement tasks like I've showed you, and then do the electrical stimulation. And at the end of everything, we then did the electrical stimulation like that, deploy those mechanical operations I showed you before. We found something really interesting from this. So first is that, let me skip past that. But basically, when we stimulate centromedian nucleus, primary motor cortex was released much more than when we stimulated any other site. This is without stimulation, brain activity and active. On axial slices, this is now during centromedian stimulation. And then when we looked at the effect of the single pulse electrical stimulation, we found that there are different shapes that you get if you measure from primary motor cortex and stimulate in the motor thalamus, centromedian nucleus, and in other motor areas. But the centromedian nucleus has a unique conduit to primary motor cortex. Based on that, we implanted him with centromedian and globus pallidus electrodes. But what's really cool, just behaviorally, as you can see here, this is him trying to perform simple task baseline and with centromedian stimulation. Centromedian was by far better than any of the others. We only discovered mechanism for why that might be the case, which is allowing thalamus to release primary motor cortex by employing these analysis techniques that we developed while delivering stimulation to support sites. So I'm sorry to have run over time. I wish I could say it was not normal for me, but I probably got that from Jeff also. Yeah, so I implanted a 4-lead system here into globus pallidus within A contact linear. So I got both the GP and GPI, and then a contact directional into center median. Anyhow, I've shown you a lot of work from a lot of people. Obviously, there's a big team, and I'm just part of it. So I'm very grateful to have them here. We actually have Lupita is here this week after this course, so we have somebody from our team here. But anyhow, thank you guys for listening to me. Really wonderful to be back.
Very much for your fascinating talk. My question is, it's interesting to know a deep background in physics. How has having a deep background in physics sort of shaped your, the way you ask questions in neuroscience? Are there any sort of shared frameworks you use or you think is led to a differentiated path you've taken to approach problems?
That's a really good question. So, I mean, obviously, there are all the tools that you have in physics. So you have all the mathematical tools. In terms of the way that you look at things, I think that physics, the idea of something called a boundary condition is there, and that is the basic understand how things behave at the boundaries, even just in physical problems. If you want to understand why going on a reed or going on a bottle, there's specific sounds that are there, that's probably some boundary conditions. Well, that exists in all sorts of different contexts. And the ability to identify boundary conditions are in the data that we measure from, I think, physics has been really helpful to me throughout my career. I don't know that you need to do a PhD. I mean, the problem sets and having to do those things sort of figured out for years during your formula probably helped me a lot. The tools are useful really now only, it's like 20 years ago. So tools are useful only that I remember that there are names of certain things that I have to look them up and figure. But it's really the understanding what boundary conditions are and applying them in biology is probably the biggest one. That's a good question. I think that's the first time anyone's ever asked me that. Thank you.
For your connectivity studies, could you change filtering so you get single cell resolution? And
So, you may wind up getting one of these papers pretty soon, a chain or a fizz, but yeah, I do a lot of single unit research also from recordings that we make in the college during AVS surgery. And it looks that technique winds up being, I showed it running just forward from the time of stimulation. You can run it forward, then run it back and isolate spikes. What's useful is you can remove the shape of the spike waveform. Actually, Stavros Anos was working on problems like this with Dr. Georgia here, but it turns out to be a really useful tool for spike removal from local field potential tracing that we measure from, particularly from motor thalamus.
One question. So for when you're measuring correlation between connectivity sites, do you avoid like losing information from having maybe a delay in phase and using dot product and not getting like maybe getting zero on what otherwise correlates, but it's just displaced in time? Do you subtract or do you move or are you doing like calculating correlation basically?
So what I think you're asking, and just so people get what he says, well, what if you have a response that looks like this and a response that looks like this? These are essentially the same thing. But because of the stimulation properties, one's delayed versus one another. So you have that. When what you need to do is shift it over. Is that what you're asking?
Yeah, do you shift it over or do you do
when applying it for the electrical stimulation, no. When applying it for because the timing is so rigid because you're really hammering the brain with the pulse of electrical current. When we apply the technique for things like visual evoked potentials, applying forward and backward, the stimulus is given out here, right? You guys have stimulus given out here, and this is the direction in time. And it's really hundreds of milliseconds later, up to hundreds of milliseconds later that you see a response. There can be trial to trial variability. So there, what you do is you still calculate dot products in the same way, but you slide them between one another, calculating the dot product for each trial to trial by trial to trial comparison. You get a profile of magnitude of dot product as a function of time, and you take the peak of that.
Okay.
So it's an extension of the technique, but we have, like, if you guys want to do that here, I can just give you code for it.